Hi everyone, my name is Dr. Caroline Morodi and welcome to today's session where we're going to look at cellular respiration. And so by the end of this session, it is expected that we'll be able to describe cellular respiration processes. And when you're talking about cellular respiration processes, we're talking about multi-step metabolic processes by which cells are able to extract energy in form of ATP from glucose and other fuels such as fructose and also galactose. And it involves three major processes, that is glycolysis, Krebs cycle, and also oxidative phosphorylation. When you look at glycolysis, it's all about converting glucose to two molecules of pyruvate. It occurs in the cytosol and it's common in aerobic and anaerobic respiration, and it will result to a yield of 2 ATPs. The next two processes which occur in the mitochondria, we have the Krebs cycle, which generates 2 ATP. And here we have the products, that is 6 NADH and 2 FADH₂, are taken up in the oxidative phosphorylation process, which result to a generation of 32 ATPs. Now, these two happen only in aerobic respiration. So let us look at glycolysis in details. When you're talking about glycolysis, it's glyco, which means glucose, lysis, breakdown, and therefore you can say glycolysis is the breakdown of glucose molecules. As we said, it occurs in the cytosol in all tissues, does not require oxygen, and there are 10 steps which are involved in glycolysis, which are divided into two groups. We have the preparatory phase, that is steps 1 to 5, and the payoff phase, steps 6 to 10. And you're going to see that there are 3 irreversible steps in glycolysis pathway, that is step 1, 3, and 10. And so let us look at glycolysis step by step. So here we have glucose, and it's converted to glucose 6-phosphate. And this is a reaction. It's catalyzed by hexokinase, and it's in a reversible reaction. And one molecule of ATP donates a phosphate groups to glucose to give us glucose 6-phosphate at carbon number 6. The activity of the enzyme hexokinase requires magnesium ion as a cofactor. We also have glucokinase, which is found in the liver, but it has a lower affinity for glucose compared to hexokinase. High levels of glucose 6-phosphate inhibits the activities of hexokinase, and this is a feedback inhibition. We see that glucose 6-phosphate is also the entry point of galactose through the process of galactolysis, which involve various steps until we form glucose 6-phosphate. It is also important to note that glucose 6-phosphate also has other fates. It can undergo glycogen formation through glycogen synthesis, and also the pentose phosphate pathway, also known as the hexamonophosphate shunt, which result in the formation of NADPH and other carbon molecules. So with glucose 6-phosphate is then converted to fructose 6-phosphate, which is an isomerization reaction catalyzed by phosphoglucomutase, also known as phosphoglucoisomerase, and it is a reversible reaction. Fructose 6-phosphate is then subjected to phosphorylation at carbon number 1, and so we have fructose 1,6-biphosphate, a reaction catalyzed by phosphofructokinase 1, and here ATP is the phosphate donor. And this enzyme, phosphofructokinase 1, requires magnesium ion as a cofactor. Now, this is the rate limiting step or the committed step of glucose to glycolysis. Phosphofructokinase 1 is subject to inhibition. High levels of ATP will tend to inhibit the activities of phosphofructokinase 1, and also high concentration of citrate will also inhibit the activities of phosphofructokinase 1. On the other hand, fructose 2,6-biphosphate, which is generated by converting fructose 6-phosphate to fructose 2,6-biphosphate, a reaction catalyzed by phosphofructokinase 2 activated by insulin will result to activation of phosphofructokinase 1, as well as AMP is also involved in activation of phosphofructokinase 1. Through the activities of aldolase, Fructose 1,6-biphosphate, which is a 6-carbon molecule, is split to 2 3-carbon molecules, that is dihydroxyacetone phosphate and also glycyl aldehyde phosphate. Each molecule has 3 carbons, 1, 2, 3, and also 4, 5, 6. Fructose, the entry point of fructose is glycyl aldehyde phosphate through the process of fructolysis. The other step involves an isomerization reaction where we have dihydroxyacetone phosphate converted to glycyl aldehyde phosphate. And therefore, at the end of the preparatory phase, we'll have 2 glycyl aldehyde phosphate molecules produced. Also important to note that we have utilized 2 ATPs in the preparatory phase as step number 1 and also as step number 2, which are both irreversible reactions. Now, the other phase involves the payoff phase, which involves glycyl aldehyde phosphate, and here we're talking about 2 glycyl aldehyde phosphate, and each will pick an inorganic phosphate and is converted to 1,3-bifosfoglycerate, and this is done through the activities of glycyl aldehyde phosphate dehydrogenase. And here we see that there is generation of NADH. 1,3-bifosfoglycerate, we then donate a phosphate group to ADP to generate ATP, and this reaction is catalyzed by phosphoglycerate kinase and requires magnesium ion as a cofactor. 3-phosphoglycerate is then converted to 2-phosphoglycerate, and here we've just moved the phosphate group from carbon number 3 to carbon number 2 to give us 2-phosphoglycerate, a reversible reaction catalyzed by phosphoglycerate mutase. And unless we then catalyze the conversion of 2-phosphoglycerate to phosphoenol pyruvate, which will donate a phosphate group to ADP, generating ATP and forming pyruvate. This is a reaction which is irreversible, catalyzed by pyruvate kinase, and it's inhibited by ATP and glucagon, but activated by fructose 1,6-biphosphate. The enzyme also requires magnesium ion as a cofactor. This is what we call the payoff phase. So in summary, this is what you said. In the preparatory phase, we have utilized 2 ATPs at step number 1 and step number 3. However, in the payoff phase, we have generated 2 ATP and another 2 ATPs, so a total of 4 ATPs. And so we can say that in glycolysis is generation of 2 ATPs because it's 4 minus 2 to get 2 ATPs. We also have generation of NADH, which will be shuttled to the mitochondria to undergo oxidative phosphorylation, as we shall see. So what becomes of pyruvate? Through pyruvate oxidation and under aerobic condition, pyruvate is transported from the cytosol to the mitochondrial matrix, where it is converted to acetyl-CoA. And this reaction is catalyzed by pyruvate dehydrogenase and requires various cofactors, including NAD+, which is derived from vitamin B3, that is niacin. We have thiamine pyrophosphate, which is derived from thiamine, that is vitamin B1, and also FAD, which is derived from riboflavin or vitamin B2. So this enzyme, which is a complex involving several enzymes, those are 3 enzymes, will result to the conversion of pyruvate to acetyl-CoA and also generate a carbon dioxide molecule. So what is the fate of acetyl-CoA derived from Krebs, from pyruvate? It enters the Krebs cycle, also known as the citric acid cycle or tricarboxylic acid cycle. And here it is linked to oxaloacetate to give us citrate. And it's a condensation reaction because we are using water to join these 2 molecules together. And this reaction, an irreversible reaction, is catalyzed by citrate synthase. Now, citrate is then converted to isocitrate via cis-aconitate. This is important to rearrange the carbon molecules so then they're able to be acted upon by the subsequent enzyme and so citrate is first converted to cis-aconitate, where we have a dehydration reaction, that the removal of water. And then the next step is hydration, that is addition of water, reaction which are catalyzed by aconitase. Isocitrate formed is then converted to alpha ketoglutarate, and here we find it is an oxidative decarboxylation reaction, because as a decarboxylation reaction, we have removal of carbon dioxide, and then isocitrate is oxidized to alpha ketoglutarate, at the same time reducing NAD plus to NADH. This reaction is an irreversible reaction catalyzed by isocitrate dehydrogenase. The next step is conversion of alpha ketoglutarate to succinyl-CoA, and this reaction is catalyzed by alpha ketoglutarate dehydrogenase, and it's also an oxidative decarboxylation reaction because they have a removal of water and also reduction of NAD plus to NADH. We also have addition of CoA, so that we have succinyl-CoA. Through the activities of succinyl-CoA synthetase, succinyl-CoA is converted to succinate, and here we have removal of a CoA. At the same time, since it's a substrate level phosphorylation, we have ADP taking up an inorganic phosphate and converted to ATP. Also, GDP can take up a phosphate group and is converted to GTP. So here we have generation of an ATP, a reversible reaction, as we said, it's catalyzed by succinyl-CoA synthetase. Succinate is then converted to fumarate through the activities of succinate dehydrogenase, and here we have a first generation of FADH₂, because two molecules from succinate are taken up by FAD to give FADH₂ and also fumarate, and here it forms a double bond. Next, through the activities of fumarate is then converted to malate by addition of water, and a reversible reaction. And malate is finally converted back to oxaloacetate through the activities of malate dehydrogenase. And here we have generation of NADH, because NAD plus we take 2 hydrogen from malate to generate NADH. So we can say we have various points in this cycle where you have generation of NADH. We have one where we have associated dehydrogenase catalyzing that reaction. We also have one where you have alpha ketoglutarate being converted to succinyl-CoA. And finally, malate to oxaloacetate will yield another NADH. And we only have one step, that is generating FADH₂, is where we have conversion of succinate to fumarate. It's also important to look at the regulatory point of the Krebs cycle. We have one where we have citrate converting oxaloacetate to citrate citrate, and where we have conversion of isocitrate to alpha ketoglutarate, and also the activities of alpha ketoglutarate dehydrogenase are also subject to regulation. When you're talking about regulation, you're talking about either activation or inhibition, and these points, which are in asterisk, are subject to activation by ADP and NAD+, and also calcium anion, especially when you have muscle contraction. Inhibition is done by ATP, NADH, and also succinyl-CoA and citrate. And this indicates that there is enough energy, so there's no need of the Krebs cycle to produce more energy. So what is the purpose of regulating the Krebs cycle? It's so that we can match the production of ATP with cellular energy demands and also biosynthetic needs. So a high cellular demand and biosynthetic needs will result to an increased Krebs cycle activities, whereas a low cellular energy demand will increase the Krebs cycle activity. It's also important to point out that there are conditions that may compromise or inhibit the TCA cycle, and these include hyperaponemia, because ketoglutarate will be used in taking up ammonia, and therefore levels of ketoglutarate will be low in the TCA cycle and therefore inhibit the cycle. Diamine deficiency, we see that alpha ketoglutarate dehydrogenase requires diamine as a cofactor. So its deficiency will compromise its activity, and this will result to accumulation of glutamate, leading to a condition known as Wernicke's disorder. Also, diabetic ketoacidosis, it's a condition seen in patients with type 1 diabetes, and it can inhibit TCA cycle by lowering the levels of oxaloacetate. Remember, we said oxaloacetate takes up acetyl-CoA and it's converted to citrate. So we have seen glycolysis result to formation of pyruvate, which enters the Krebs cycle. We have seen it also lead to generation of NADH, which we shall see what becomes of NADH. When you've moved to Krebs cycle, you have seen it is producing FADH₂ and also NADH. And so we want to see what is the fate of these high energy electron carriers. And so this brings us to oxidative phosphorylation, which can be divided into two processes, that is the electron transfer chain and also chemiosmosis. And so when you're talking about electron transfer chain, it's just a series of redox reactions, which is composed of 4 complexes labeled 1 through 4. We have complex 1, which is NADH dehydrogenase. Complex 2, we have what's called succinate dehydrogenase. Complex 3, that is cytochrome BC1 complex. And finally, complex 4, which is cytochrome C oxidase. So we shall see that electrons are passed from one component to another until complex 4, where we have oxygen as a final acceptor of electrons, producing water. So let's look at that in details. So we look at the electron transport chain. We have complex 1, which is the NADH dehydrogenase. Here, NADH, which is carrying electrons, will pass the electrons to complex 1, and it is oxidized to NAD+. As the electrons are transferred to complex 1, complex 1 is charged and pumps 4 hydrogens or 4 protons from the matrix into the intermembrane space of the mitochondria. FADH₂ will enter the electron transport chain FADH₂ will enter the electron transport chain through complex 2, and here it will donate its electrons or pass its electrons to complex 2, and it's reduced to FAD. Here there's no pumping of hydrogen from the matrix to the intermembrane space. So the electrons in complex 1 and complex 2 are then passed to cytochrome Q or coenzyme Q. They are passed to coenzyme Q, which is a lipid-soluble electron carrier, and passes the electrons from complex 1 and 2 to complex 3. So when complex 3 gets the electrons, it is charged and pumps 4 hydrogens or 4 protons from the matrix to the intermembrane space. Thereafter, it transports the electron to cytochrome C, which is a small soluble protein, which then will transfer the electrons to complex 4. So at complex 4, that is the cytochrome C oxidase, it is charged and pumps 2 instead of 4 hydrogens or protons from the matrix into the intermembrane space. Now the other 2 electrons will be used in formation of water. And here we'll see that the electrons that have been transferred to complex 4 are taken up by oxygen and reduced to water. So as we have said earlier, oxygen is a final acceptor of these electrons, and it is reduced to water. So that is the electron transport chain. And then we move on to chemiosmosis. And here, when you talk about chemiosmosis, we're just talking about maintaining an electron gradient. Now, remember we were pumping hydrogen or protons from the matrix to the intermembrane space. And so we have a higher concentration of protons at the intermembrane space. And so to maintain a gradient, the protons will be taken back to the matrix. And in so doing, it will activate the ATP synthase. So the hydrogen ions are being pumped from the intermembrane space through the ATP synthase, activating it, and therefore it will be able to convert ADP and an inorganic phosphate to ATP. And that is what we are calling chemiosmosis. So the electron transfer causes a concentration gradient where we have higher protein concentration in the intermembrane space. And as they are moving back to the matrix, they will activate ATP synthase, which will therefore be able to convert ADP and inorganic phosphate to ATP. This is important to note. When we are transferring electrons from NADH, we are pumping 10 protons. Let's see. So here we pumped 4 protons. Here we pumped another 4 protons. And here we pumped 2 protons. So those are 10 protons pumped from the matrix to the intermembrane space when electrons from NADH are passed from one complex to another. When it comes to FAD, it will pump out, let's see how many hydrogen atoms or ions will be pumped out. That is 4 and another 2. That's a total of 6 protons pumped when the electrons from FADH₂ are passed across the complex. Remember this, 1 ATP is synthesized when 4 protons are taken back from the intermembrane space into the matrix. What of 10 protons? How many ATPs will it generate? That is 2.5 ATPs. So that is why we say one NADH molecule will generate 2.5 ATPs, whereas one FADH₂ molecule will generate 1.5 ATPs molecule. The oxidative phosphorylation is also susceptible or can be regulated. And one aspect that regulates oxidative phosphorylation is substrate availability. So if you have ADP and inorganic phosphate, which is a key limiting step, it will allow oxidative phosphorylation to take place. It will be activated. Presence of also NADH and also FADH₂ are also activator of oxidative phosphorylation. Oxygen availability also regulates oxidative phosphorylation because we've seen it's a final electron acceptor. And hypoxia will limit ATP production because we have seen that oxygen is very important in this step, especially in the last complex 4 where we have oxygen accepting the electrons. And so this is what we call oxidative phosphorylation. It's composed of both electron transfer or electron transport chain and also chemiosmosis, which involves ATP synthase. So we have seen that all the products of glycolysis, that is NADH and ATP, we've seen the products of Krebs cycle, that is NADH and also FADH₂. At the end of the process, that is through oxidative phosphorylation, they're able to generate more ATP as required by the human body. It is important to note that there are other inhibitors of the electron transfer chain or oxidative phosphorylation. One, we have rotenone. Rotenone is a plant-derived insecticide and it blocks the transfer of electrons at complex 1, and therefore there's no pumping of hydrogen ions or protons. This may affect ATP synthase. We have antimyosin, which is a natural occurring compound. It blocks transfer of electrons from coenzyme Q to cytochrome C via complex 3. And this will hold proton pumping at complex 3. And ATP production will cease. Cyanide is a very important but highly toxic chemical that exists as gas, and it inhibits complex form because it binds the heme ion in complex form, and so there is no transfer of electrons to oxygen. This will halt the entire chain, and protein gradient will collapse, and so no ATP synthesis will take place. Others include the uncoupling agent and also oligomyosin, which tend to inhibit ATP synthase. And so with that, we've come to the end of this session where we have looked at the three processes that are known as the cellular respiration, that is glycolysis. We have seen glycolysis will eat two ATPs and also NADH. We looked at Krebs cycle. We have seen the products include NADH, FADH₂, and ATP. And finally, we have looked at oxidative phosphorylation, where we have seen that the products of glycolysis and Krebs cycle, that is NADH and FADH₂, are able to enhance formation of more ATP as required by the human body.